Welcome back, friends. We've been talking about digestive system and the different parts of digestion in human body. In this video lecture, we'll be talking about the digestion and absorption that is going on in our body. We've been looking at different anatomy of our digestive system. We saw the different types of control and regulation of the digestive system. Now this is the time to actually understand how the digestion and absorption take place and what are the different regions for this process. Now chemical digestion involves breaking down of the food with the help of enzymes, and those enzymes will break down macromolecules into small fragments and nutrients. Once those small fragments are generated, then they are absorbed in the GI epithelium cells. In this lecture, we will be discussing the action of digestive enzymes involving amylase, proteases and lipids, along with the function of bile salts. We will also describe the transepithelial transport of simple sugars and amino acids. And thirdly, we will be discussing the emulsification and chemical digestion and absorption of lipids, which is a slightly unique compared to the carbohydrate and protein digestion and absorption. Let's talk about the chemical digestion process that actually take place in mouth from the beginning till the end in the intestine. The ingested food first is broken down mechanically in the mouth. That's known as the mechanical digestion. Once they are broken down into fragments, into smaller pieces enough to be swallowed, then they will be taken inside the esophagus. Through the esophagus, they ultimately take entry into the stomach. Now inside the stomach, there are two types of digestion take place. One is a little bit of mechanical digestion and a little bit of chemical digestion as well. The small particles are then mixed with acids, which is hydrochloric acid secreted by the stomach cells, and then this acid will help and allow to break down some protein fragments inside. So this is a kind of protein digestion begin in the stomach. Now the major nutrients must be broken down enzymatically in sequential stages in the small intestine to finally produce the fragment portion of all carbohydrates, proteins and lipids. Now if we look at here, there are chain of all these things, polymers of carbohydrates, lipids and proteins. The polymer of carbohydrate example is starch. When broken down, it produces unit sugar, that is glucose. Similarly, we have protein molecules. When you break them down, it produces amino acid. And we have fats. When you break them down, we get fatty acid and triacylglycerol as a unit component. Here you see the different list of enzymes that are involved with the substrate breakdown into small fragments. For example, breakdown of starch is mediated by amylase and it's secreted both one in mouth and another from the pancreas, and in both of them break them down and form maltose and glucose. Proteases act on stomach and small intestine, break down protein into amino acid. Lipase acts in the small intestine, in the duodenum, break down fat into fatty acids and glycerol. Now all carbohydrates must be digested enzymatically into the monosaccharide components. That's the first and foremost thing. Whatever carbohydrate we are dealing with, it must be broken down into the monomers. Now starch is digested into the monomer glucose, while sucrose is digested into the monomer glucose and fructose, and lactose is digested into glucose and galactose, while maltose is digested into 2-glucose. This should be produced. Proteins all the time should be converted into amino acids. Now the amino acids that they can produce by breaking down protein could be like a dipeptide breakdown of this protein into 2 peptides or 2 amino acids together, or they can form tripeptide, so 3 of the amino acid can be linked. And most of this fat is broken down into fatty acids and triacylglycerol that we talked earlier. Now there are some also non-polar products in the digestion, and that can be absorbed by simple diffusion process, which is a passive diffusion through the phospholipid bilayer of the intestinal epithelial cell. Okay, and most of the polar substances are absorbed using active transport, or in some cases passive carrier mediated transport through the cell membrane. So we have the channel proteins through which it can take entry. Fats or non-polars can easily take entry through the membrane because that is mostly hydrophobic. Let's first talk about the carbohydrate digestion in the mouth. It's the first start point, and the carbohydrate digestion begins in the mouth. This is the type of chemical digestion that we are talking about. Normally, mouth is involved with two types of digestion. One is mechanical, second is chemical. In the mechanical digestion, teeth is involved to break and grind all those food into smaller particles, and then they will start mixing these food particles with saliva. Saliva contains amylase, and the amylase can break down starch into fragments. Amylase break down starch into following components. It can break the starch into maltose and maltotriose and some amount of dextrin. So maltose is normally a disaccharide, maltotriose is a trisaccharide, and dextrin is a kind of branched fragment that is generated. As you see, the starch is a hugely branched molecule. It's been broken into all these smaller fragments there. Okay, but when all this mixed amylase and all the chemical agents are transferred from mouth through the esophagus into the stomach, inside the stomach acid deactivates this salivary amylase. So the function of salivary amylase is only active in the mouth and when the food is moving through the esophagus. That's the only part salivary amylase is active, but once it's mixed, it is enough to break carbohydrate down because it takes less effort to break and digest carbohydrate. The second thing is about the protein digestion, and the protein digestion begins in the stomach. So most chemical process, like normally the process of protein digestion occurs in stomach because stomach releases acid and another enzyme known as pepsin. Actually, pepsin require acidic environment to act and break down protein components. That's why they need to release acid there, and they release hydrochloric acid in the stomach. While the pepsin will start activating, it breaks the peptide bond between the tyrosine and phenylalanine. Remember, it breaks the bond of any amino acid sequence or repeated peptide between tyrosine and phenylalanine into smaller amino acids, and they produce peptide fragments, and that contains few amount of an amino acids. So this is a larger protein broken down by pepsin into smaller fragments of amino acid chain. Stomach also responsible for a little bit of mechanical digestion, where the stomach has its own peristaltic waves generated that can break and grind foods a little bit inside the stomach. Now if we talk about the process of digestion in the small intestine. Small intestine is the region where most of the chemical digestion take place, and it is also involved with the absorption of all those nutrients inside. Pancreatic digestive enzymes alone are much sufficient to digest carbohydrates and proteins that are present here in the small intestine, because pancreas secretes all those proteolytic enzymes. Example, trypsin, chymotrypsin. These are the examples. It produces pancreatic lipase. They also provides pancreatic amylase. Pancreatic lipase is essential for the bulk of fat digestion, and for example, cellulose and other type of plant complex carbohydrate cannot be digested by the humans because we don't have any of the enzyme to break them. That's the reason we cannot eat grass, because the most of part of the grass is made with cellulose, which is the plant cell wall component. So indigestible plant polysaccharides serve as an important source of dietary fiber, which is also required for a proper movement of the bowel through the large intestine. And certain fiber type help the movement of the stool through the colon. Now if you look at here, the vegetables and the fruits contain this amount of dietary fiber, but they mostly contain valuable vitamins and minerals as well. So all those vitamins and minerals are absorbed in the small intestine. And most of the dietary salt that we take, let's assume these are all dietary salts, those things are absorbed by the small intestine by either the transcellular or paracellular transport mechanism in the small intestine. So what are those type of scenario? In the transcellular transport of water, water is linked to active transport of sodium via sodium potassium pump. So water is taken and dragged inside with the help of that sodium potassium pump. Sodium is actively pumped out through that process. So let's say this is inside, this is outside. The sodium is dragged out, potassium is dragged in. Once the sodium is actively pumped out of the digestive tract at the basolateral membrane of the epithelial cell, the sodium then moves passively from the GI lumen into the epithelial cell through the ion channels, which is a sodium glucose co-transporter channel. So once sodium is moving inside the cell, glucose is also moving inside the cell, and then chloride follows the sodium. It's through the electrochemical attraction process, chloride also start to flow inside, and then as a lot of ions are moving inside the cell, the concentration of ions are increasing inside the cell, and to keep this concentration of inside, the ion concentration inside and outside constant in an equilibrium, water start to passively inside those cells via osmosis. That's the overall idea of absorption that is taking place. As you see here, we will be looking at this process, that this is the first part of the digestion in small intestine, mediated for the carbohydrate digestion and absorption. So here come the pancreatic amylase that continues to digest all those large fragment of carbohydrates into smaller fragments, and actually these enzymes work in the duodenum, which is the start point of the small intestine, and there are intestinal cells known as the brush cells. So this is known as the brush border of the cells. Okay, for the digestion, these brush border enzymes complete the process of carbohydrate breakdown into monosaccharides, and in this case normally the glucoamylase break down the maltose. So there are different enzymes. Glucoamylase break down maltose and maltotriose, so those things are broken down by glucoamylase. Dextrinase breaks down dextrine. Okay, I'm not writing it here. So dextrine is broken down by dextrinase. Okay, any enzyme you'll see ase will be the enzyme to break it down. For example, to break down sucrose, we have sucrase secreted there. For breaking down lactose, we have lactase. So these are all carbohydrate breakdown enzymes that are secreted there and work there. Now most nutrients that are absorbed mostly by the transepithelial transport. So what is that process? You know, there are different transporters that are present in the epithelial cell membrane. Normally it could be primary or sometimes secondary active transport. In the secondary active transport is which the glucose and galactose are co-transported. Glucose and galactose are co-transported inside the cell together. Fructose enters the epithelial cell normally on its own via the own transporter that we see. It's a fructose specific transporter, but glucose and galactose, they transfer the same time at a specific region. And we also have glucose sodium co-transporter, which also helps to move glucose using the sodium ion concentration gradient there. And once inside the epithelial cell, all the monosaccharides that take entry, monosaccharides exits the basolateral side of the cells in the same type of transport molecule via the facilitated diffusion, because that will be possible to move from there into the capillaries and spread through the body and rest of the area of the body cells. Here you see the process that once this glucose and sucrose and fructose, all these things are entered inside the cell, they will be transported out into the capillary by the process of facilitated diffusion, where there is a carrier protein required to transfer those monosaccharides into the specific place of the capillary. Now the second part of the digestion in the small intestine is the digestion of proteins. And if you look at the digestion of proteins here, if you see, there are small intestine, because in the smaller intestine we have the protease enzymes released from pancreas, a variety of proteins and protein breakdown products that are formed. For example, trypsin. Trypsin is an activated form of trypsinogen, while chymotrypsin is an activated form of chymotrypsinogen. Carboxypeptidase is an activated form of procarboxypeptidase. They are all the major pancreatic proteases that approximately work at pH 7 for the process of protein digestion. Trypsin and chymotrypsin, these both two, trypsin and chymotrypsin, split the protein into smaller peptides and some of them into single amino acids even. So only single amino acids can be generated, while carboxypeptidase splits single amino acids into carboxyl end of the protein and start breaking down amino acids from the terminal. So carboxypeptidase work after the activity of trypsin and chymotrypsin. Okay, and here you see there are also brush border cells that are present in the epithelium, right? So these are the brush border enzymes known as aminopeptidase and dipeptidase continues the process of digesting those fragmentized amino acids so that we can absorb that. So once they produce single, either they can produce single amino acid or produce di amino acids, or they can produce tri amino acids or tripeptides that can be absorbed through the small intestinal cells. A number of different amino acids are co-transported into the luminal side of the epithelial with the help of sodium and via secondary active transport. So again, sodium can help, the sodium gradient across the cell, let's say a lot of sodium gradient is generated here. Sodium gradient across the cell can help to bring some of these peptides as well, dipeptides and tripeptides as well, to bring inside the cell as a co-transporter mechanism. Now most of the proteins absorbed as dipeptides and tripeptides, as we talked earlier, and which are further digested into single amino acids inside the GI epithelial cells. So once you get a dipeptide or let's say tripeptide inside this epithelial cell has a lysosome that is filled with proteolytic enzymes. Once they release that, it can break those tripeptides and dipeptides down into smaller single monopeptides. Now if we have more hydrophobic amino acids, and those amino acids tend to leave the basolateral side of the epithelial by normally simple diffusion mechanism into the capillary there. Now if you have more hydrophilic amino acids, they want to exceed the GI epithelial by the process of facilitated diffusion. So in this picture it's shown that mostly these amino acids are hydrophobic, so they can easily transfer themselves through the membrane into the blood vessel there. But if it carries more of a hydrophilic one, it should have a carrier protein embedded into the cell membrane that will help to move the amino acids into the place. Now it's time to talk about the digestion of fat in the small intestine, and fat digestion is kind of unique. It's slightly different compared to the digestion of digestion of carbohydrate and protein, because fat digestion takes a lot more time. Most of the dietary fats are triglycerides, which are made up of 3 fatty acids, which could be like short, medium and long hydrocarbons, and each of them are bonded with glycerol, okay, with the help of ester linkage, those fatty acids. So there is a glycerol and hydrocarbon tails are attached to it with the help of ester linkage in the middle. Now all of this actually most of this digestion occurs in the duodenum of the small intestine. Mostly the process of chemical digestion that conveys in the small intestine occurs in the duodenum. and the absorption takes place in the further downstream of the small intestine. Okay. Now there are two mechanisms act to increase the surface area of the fats and subsequent chemical digestion. One event is the release of bile, and second event is the segmentation, which is a physical event as well. Segmentation in the small intestinal wall. Segmentation will help the fat to disperse into the aqueous layer, like the oil and the vinegar. If you see here, this is the oil and vinegar and how it's dispersed. If you have a segmentation, like as you see, there is a segmentation here in this specific bottle. Due to this kind of segmentation, we have a different way of separating the oil from the rest of the parts. So most of the molecules that are present on top are the oil, are the fat, and most of the aqueous solution is residing in the bottom. Now if you do the same thing with other kind of structures, it will be kind of hard to achieve, but if you have segmentation, it helps. And the second thing, bile salts attach to the smaller fat droplets and prevent them for attaching with themselves and form a large fragment. So it will keep this smaller fragment of fat separated, and we call it as an emulsification. Now as the bile salts are amphipathic in nature, that means they have a hydrophilic as well as a hydrophobic side, they can surround the small lipid droplets. So let's say this is a small lipid droplet, and the bile salts are surrounding that lipid droplets and separating it to the rest of the other lipid molecules, okay, and prevent them from coming together. So in this case, they increase the total surface area of each of the lipid fragments by separating them into smaller fragments. So increasing in the surface area of the lipid helps for more efficient hydrolysis of these lipid molecules by the pancreatic lipase enzyme. So pancreatic lipase will now act, right? So lipase digests triglycerides into monoglycerides and free fatty acids. So what it will produce? It will produce the glycerol. That backbone will be there, the glycerol backbone, and we have all those hydrocarbons released. Now that backbone can also be broken into smaller fragments. So it's a triglyceride, it can break them down as well. Now bile salts also surround these monoglycerides and free fatty acids as well to form what tiny droplets known as micelle droplets. It's kind of like 1 million times smaller than the emulsified fat droplet, very very small. So even after the digestion is done, still the bile salts keeps like circulating, keep covering those fragmentized portions, okay. And why it is required? Let's say here this is the large lipid droplet, mixing of bile salts, breaking them down into smaller fragments known as micelle, and micelle is formed even after the breaking of the fragments. We have those bile salts to cover them up. That's what we call micelle here. Now once we form the micelle, it comes close to the luminal membrane. This is the luminal membrane that we can see, close to the luminal membrane of the epithelial cells here, and they release their monoglycerides and free fatty acids inside the cell via simple diffusion through the lipid bilayer. That's what we can see here. So here we have the smaller fragments of fatty acids and monoglycerides that can be easily diffused through the core area of the cell membrane, because most of the part of the membrane is actually made with hydrophobic region. So now this is one of the way of delivery. Or what happens, some of this micelle can directly interact with the cell membrane that ultimately release triglycerides and fatty acids inside the cell. Once we need to transfer that triglyceride to some distant area of the cell for the process of metabolism, that triglyceride and those fatty acids are further coated with proteins, and then we bring them to the capillary or any lymphatic system if we require. And those fragments which are coated right after the process to transport are known as chylomicron. Chylomicrons are the dressed up fatty acids and glycerols that is transport ready, okay. And this chylomicron exit the basolateral surface of the epithelial via exocytosis process, and then they will enter the lacteal lymphatic system here. So this is the lacteal lymphatics. Through this lacteal lymphatic system they will be migrated. They don't actually enter into the capillary, they go to the lacteal lymphatic system. So that's how the release of fatty acids are made in the digestion in the small intestine. Now the last thing that I want to talk about is the absorption process that take place in the large intestine, because large intestine is not responsible for the chemical digestion, neither it's a function of secretion. So the only function it plays is the absorption of the nutrients and absorption of water and absorption of some amount of ions and vitamin K. So there's a kind of symbiotic relationship between some bacteria that resides in our large intestine. Some of the example is E. coli, that's a friendly bacteria that is present in our large intestine that helps to generate vitamin K that is absorbed by these large intestinal cells, as well as large intestinal cells absorb a lot of water to make the feces much more concentrated to be released outside the body. So here the byproducts of those colic bacteria metabolism include small amount of vitamin B complex and a huge amount of vitamin K. Both are absorbed in the intestine. And here vitamin K is very essential for producing blood clotting proteins. So without vitamin K the blood clotting pathway can be compromised in our body. Now the colon absorbs the small quantities of salt as well, as you can see here. This is lumen of colonic crypt and this is the interstitial fluid. So what we are going to do, it can take some amount of sodium and chloride ion inside. Chloride ion enters into the lumen with the help of CFTR channel proteins present. Sodium is reabsorbed, and it also, as they start to build up a lot of sodium and potassium inside the cell, it will automatically allow the water to backflow to balance the osmotic pressure. That's the whole idea of the absorption there. It's not a primary content of absorbing water, but as a lot of sodium and chloride is being absorbed to balance the equilibrium of the concentration of the ions, water flow in the osmosis pathway. That in a sense is the absorption process that takes place in the large intestine. I hope this video helped you to understand about the digestion and absorption that is going on in our body. So if you like this whole series of video about the digestion, please hit the like button, subscribe to my channel to get more and more anatomy and physiology videos like that. Thank you.